Okay, so obviously your review questions are here. You can start thinking about these, and we're going to talk we have a couple announcement kind of things, and then we'll finish chapter 27. Okay, one more minute. We have a lot to get through today. Okay, so what's the difference between a positive sense single-stranded RNA virus and a negative sense single-stranded RNA virus? Anna.
I said for positive sense single-threaded RNA, it's direct translation and an example I put COVID in it.
Okay, so positive sense is directly translated. COVID is an example of it. Lots of examples of it, but that's one. How about a negative sense? What's a negative sense? What has to be done? So it brings its own enzymes to make like a complementary mRNA So it can't be directly translated, it has to make its own complementary mRNA to be translated. What are some examples of a negative sense single-stranded RNA virus? This is class, I was confused 4 and 5. This is class 5.
Rabies, mumps, measles, flu.
Rabies, mumps, measles, flu. All of those are examples. The other category in there of single-stranded RNAs, single-stranded RNA category 6, class 6, which is HIV. That's the one that brings reverse transcriptase in and makes DNA before anything is transcribed and translated. Okay, and what are 2 characteristics of all prokaryotic cells? There's more than 2, but there's a lot. Just say them. Okay, never mind. You can't just say them. There's too many people saying so I can't hear. What did you say, Athena?
Genetic information, ribosomes, cell membrane, cell wall.
Okay, they all have genetic information. They all have a cell wall. They all have a cell membrane. They all have ribosomes.
There's no membrane-bound organelles.
No membrane-bound organelles. So the nucleus, there's no nucleus. There's a nucleoid region where the genetic material is located, but there's no actual nucleus. Nothing to keep the DNA separate from the rest of the cytoplasm. I don't remember if I said anything else. Oh, the cell wall in bacteria is made of peptidoglycan, which is a different chemical than when we talk about a cell wall in a fungus or a cell wall in a plant. And that's kind of where we left off. We were in the middle of a slide about bacteria. We do have a fair amount to get through, and I think we can do it. We'll see how it goes. Regardless of what happens in class today, we're going to start chapter 28 on Monday. Okay? Chapter 28, we're getting into eukaryotic cells, and we're going to talk about protists, which is possibly the weirdest category of organization of living things ever. It's just like the random assortment of stuff we don't know what to do with. Test is next Friday. In terms of your bacteria, so I looked at plates this morning. They're not doing what they're supposed to be doing, and I don't know why. It's weird because some plates are growing right and some plates are not growing at all.
Are you talking about the smaller ones?
Yes, the small ones. So if you're in Wednesday lab, I pulled out your red mannitol salt agar plates and put them next to the incubator. Thursday ones, we can probably pull them out. I just glanced at them and I didn't pull them out yet. So I'm going to pull all the plates and put them next to the incubator. If your bacteria didn't grow for the Piccolo transformation lab part, for the little plates, look at one that did and record those results. I'm trying to figure out why. Again, it's never really not worked for me, and so I would like to say that it's the bacteria or the agar, which would be my fault, but the fact that some of them grew is inexplicable to me. So I'm still working on a hypothesis for this. So just know that if yours didn't grow, find a set of plates that did grow and make sure you're looking at those under the UV light, not just looking at them with your naked eye. Okay? Questions about that?
When are we supposed to be looking at those?
By Monday afternoon. And again, actually the ones that didn't grow, I'll probably leave them in the incubator to see if they will grow. Again, I don't know what's going on. It's weird to me. If all of them hadn't worked, I could blame it on the agar or I could blame it on the bacteria. But the fact that a couple groups did work and some of them didn't work, I can't explain yet. I'm going to work on that. Okay, so let's talk about bacteria. So we left off. Oops. Nope. Why is it not doing anything for me here? I want the picture, that's why I'm, okay. That's all. Okay, we left off, we kind of talked about the plasma membrane looking similar to a eukaryotic cell membrane. It's a phospholipid bilayer, it has proteins in it, selectively permeable, all those kinds of things. We started talking about the cell wall, and we were looking at the difference here between these 2 cell walls. And we said the top one was which kind of bacteria? This is gram-positive, and this is gram-negative. Okay, so the cell wall, no matter which kind of bacteria it is, has a function of providing structure for the cell. It provides protection from the cell. So if the cell gets an environment, a hypotonic environment where fluids are coming into the cell wall can prevent the cell from bursting. So it has the same function in all of them. But you can see it's structurally different in gram-positive versus gram-negative. But again, both of them are peptidoglycan, which is unique to this kingdom, unique to the bacteria kingdom. Peptidoglycan is a polymer. What is a polymer? What does that mean? It's a repeated subunits, lots and lots of subunits together. It's a repeated polymer of subunits of sugar, and those subunits are cross-linked, so they're connected by proteins. So peptidoglycan is a sugar polymer with cross-linked, it's cross-linked by proteins to hold it together. And you're not really seeing that in this diagram. And we've already kind of alluded to and talked about, in fact, in lab, that gram-positive and gram-negative have different cell walls, and that's why they stain differently. This is what gram staining is what we usually do to help identify something. That's the first step in identifying a bacteria, is identifying what kind of cell wall does it have. Is it gram-positive or gram-negative? That's going to be the first thing you do anytime you identify a positive, a happy bacteria, a bacteria that's not harmful, a pathogenic bacteria. What we care about is it gram-positive or gram-negative, because we're going to treat it differently. And we'll talk about that in a minute. Again, this gram-negative has these two cell membranes, so it's got an inner cell membrane and then it's got an outer cell membrane. They have lipopolysaccharides on them. Lipo meaning lipid, polysaccharide meaning many carbohydrates, many sugars, many monosaccharides. So those little markers on the cell membrane, on the outer membrane, actually can be toxic to cells. They can produce toxins. They themselves can be toxic. And so that's what makes gram-negative a little worse than gram-positive, which we'll talk about in a minute. The next slide we don't really have to talk about too much because we've already done this in lecture. We've done the gram staining process. So you know the primary stain is what? Crystal violet. Crystal violet stains everything purple. We add grams iodine, which is the mordant. That makes the crystal violet iodine complex, which is a bigger molecule. Okay, then the next step is the important step. This is what allows it to differentiate between the two kinds of bacteria because they respond differently. The decolorizer, the alcohol decolorizer, is going to wash the primary stain out of gram, which one? Gram negative. So here, our bacilli are gram negative. Alcohol dissolves lipids. Alcohol is able to dissolve lipids, so the alcohol can dissolve that outer membrane, that outer lipopolysaccharide layer of the gram negative is dissolved by the alcohol. And then because the peptidoglycan layer is thin, it can just wash out that primary stain. The gram-positive cells, which in this case are the cocci ones, the gram-positive cells, it kind of tightens up. It dehydrates the proteins that are in the cell membrane and the cell wall and kind of tightens it up so that the primary stain can't get out. So it has an opposite effect. So it washes it out of gram-negative and it leaves it in gram-positive. Then we add safranin or safranin, people say it different ways. And the gram-positive cells aren't going to take it up because they're already purple, you're not going to see it. And the gram-negative cells are going to take it up because they are not stained anymore, so they will appear pink versus purple. And we've seen this in lab. But here's an example of a slide that has a nice gram-positive cocci and a gram-negative what? Bacilli. So this could be like E. coli and Staph aureus. I don't know if it is for sure or not. So I kind of alluded to, and we're going to talk about the fact that we treat these differently when we're looking at the cause of an infection. We're going to treat them differently because of the composition of their cell wall. So gram-negative bacteria have that lipopolysaccharide layer, that extra outer membrane. Again, this is toxic to our cells. So your body can respond to this with fever or shock. That extra membrane also gives it more protection. It gives it more protection from the immune system. So it makes it harder for your body's immune cells to engulf and take in the bacteria and kill them. So it makes it harder to phagocytize the gram-negative bacteria. So it offers protection from that. It also, so protection from the immune system. It also makes them more resistant to drugs. So they are not as responsive to antibiotics, which is kind of like our first, that's how we treat bacteria. So they're more protected from the immune system because of that outer layer, but they're also more resistant to antibiotics. One of the reasons that they're more resistant to antibiotics and gram-positive are more susceptible to antibiotics is because antibiotics, some antibiotics, the mechanism that antibiotics work with, they can actually interfere with the cross-linking. So we said that the peptidoglycan is the saccharides, the polymers of saccharides cross-linked by proteins, so held together by proteins, and antibiotics can interfere with that process of cross-linking, which makes the cell wall unstable, which makes the bacteria die. So a bacteria that is gram-positive, that has a really thick peptidoglycan layer, if we can interfere with cell wall synthesis, that can kill the bacteria. So something like penicillin is an antibiotic that specifically targets cross-linking of proteins in a peptidoglycan. You don't have peptidoglycan in your cells, so it's not going to affect your cells. It's only going to affect bacterial cells. So that's a way that we can target prokaryotic bacterial cells versus human eukaryotic cells. So it can interfere with cross-linking. Some of them actually will interfere with synthesis of the cell wall in general and other ways, but penicillin is the one we always use to talk about as an example for this.
So I think it was last lecture you mentioned MRSA, the methicillin resistant.
Yes.
Will penicillin still work against it, or
So
what's the...
for MRSA, they use vancomycin. I don't know if Staphylococcus aureus is susceptible to penicillin or not. So there are broad spectrum antibiotics which will affect a lot of bacteria, and then there are narrow spectrum antibiotics which only affect some bacteria. So I don't know if Staphylococcus aureus is susceptible to penicillin in general. Penicillin tends to be a broad spectrum, but I know they use vancomycin for MRSA. They use penicillin for strep throat and lots of other things, but I don't know for MRSA specifically if they could use penicillin, but I know vancomycin is the current one that they're using for that. And I only know that because I have MRSA in my nose. Okay. Gram-positive versus gram-negative. Okay. Okay, we're going to move on to some other structures that bacteria can have. And these are not necessarily true of all bacteria, so these are more, again, thinking about the fact that bacteria are really diverse and adapted to lots of different environments. Some bacteria have these, some bacteria don't have them. So this is not true of all bacteria, and actually gram-positive and gram-negative is not true of all bacteria, just most. If you take micro, you can learn about all the exceptions to that. Okay, so some bacteria can produce a glycocalyx layer. So glycocalyx is, again, it's a layer of polysaccharides that's really sticky. It can be made out of proteins too, but that's less common. It's more common for it to be made out of polysaccharides, and it makes it sticky. I think of it like corn syrup kind of stuff. Like corn syrup is really sticky and gross. Okay, and a glycocalyx can take two forms. It can be a capsule, or it can be a slime layer. And that has to do with the level of organization. So a capsule is an organized layer of polysaccharides, and a slime layer is not an organized layer. So it sticks more loosely. A slime layer is looser than a capsule layer. The advantage of having a glycocalyx that's sticky might be what? What would be an advantage of being sticky if you're a bacteria? You stick to stuff. So you might stick to a substrate, like something that's not living. Again, it also makes them more resistant to being phagocytized by the immune system. So advantages are you can also stick to each other. And that's how things like biofilms can form with stickiness of bacteria. So this is a picture of bacteria that have a capsule. So you can see inside there's that little purple dot. That's the bacteria. Then there's that clear layer around it. That's the capsule. And then the background is just stained pink too. So all those bacteria have that clear layer around it. That's a capsule. And they don't stain. They're anionic. They have a negative charge to them, so they don't stain with the stains that we use, which is why they end up looking clear. So some bacteria can make these, and they make them sometimes, and having the capsule or the glycocalyx layer makes them infectious. And if they don't have it, they're not infectious. Strep pneumoniae is not infectious without its capsule. Anthrax can form a glycocalyx layer. It makes it infectious. Speaking of anthrax. Oops, sorry. I need the picture. And now I need to erase that. Another structure, again, that some bacteria can form is an endospore. So endo meaning what? Inside. So it's within a cell. It's a spore, so it has a really thick coat on the outside that makes it resistant to almost everything. Okay, so bacteria are going to form an endospore if conditions are unlivable for them. So if things get bad and they can't handle life, they can form an endospore, and that's going to contain the genetic material needed to become a healthy, living vegetative cell again if things get good. Okay, so this top picture shows you bacteria. This is probably B. cereus. It could be anthrax. It's a bacillus. You can see the red cells are what we call vegetative cells. So those are like metabolically active. They're alive and metabolizing. And then inside the cells you can see little green dots. Those green dots are the endospores. And again, only some bacteria can form these. So if you stain bacteria using a spore staining method, you can tell if they can make endospores or not, and that can be useful in identification. If things get really, really bad, they release the endospores to make free spores, and those can go out and live forever and ever and ever and ever and ever and ever until things get good, and they can, maybe not forever and ever, but they can live for extended periods of time. In drought conditions, they can survive all kinds of things. You don't remember probably because you were not alive, or you were so small that you don't remember, but around the time of September 11th, when the whole terrorist attack happened, there was a guy who started mailing people anthrax spores in the mail. Because it can survive, it doesn't need anything to survive. Spores can survive in pretty much any condition. And so he started mailing people anthrax spores. And people died of anthrax because they inhaled the spores, and then in their body, the conditions were right for the spores to become metabolically active vegetative cells again, and they got anthrax and died. Yeah, it kind of was, yeah. It wasn't like a really like it just was a powder. He just mailed it as a powder, and people would open the envelope, and just opening the envelope would release enough spores. And
would you even have to open the envelope?
you would have to open the envelope. I don't think well, I shouldn't say that for sure. I don't think anyone who died from it didn't open the envelope. But I mean, they would be small enough to fit through the pores in the envelope, but you would have to get enough still that you would inhale. That would be a problem. but like people were terrified that they were going to get anthrax in the mail. It was a really weird time in the world, like now. So this bottom picture is showing you the endospore. That's the endospore within the cell. Again, it has a really thick layer on the outside that's protecting it from the conditions that are not good. Next one is a Fimbriae. I'm going to get the picture. So again, some bacteria can make these, some cannot. Fimbriae are these hair-like structures. This is a like colorized electron micrograph of a bacteria Fimbriae. They wouldn't look like this under our microscopes. So they are hair-like structures. There's usually lots of them. And they're formed, they can use them to stick to stuff. It reminds me, I don't know, Spider-Man. That's what I think of when I see it. Yeah, bacteria man, bacteria spiders, I don't know. That's getting creepy. They can stick to a surface or they can use them to stick to each other. This is a picture of Neisseria gonorrhea, which causes gonorrhea. It sticks well to body cells because it has these Fimbriae. They're shorter and there's lots of them, unlike the next one, which are Pili. Pili tend to be longer, and I thought there's a picture of them in a slide or two. Pili are appendages that are used to pull bacteria together so they can transfer DNA. So the Fimbriae are for adherence. Pili are for DNA exchange, but sometimes they're also used for movement. They tend to be longer, and there aren't as many of them, but both of these are made out of proteins, and they have the same protein makeup. So they're structurally the same in terms of protein. It's just number and length that makes them different. So we said Pili can be used for movement, but what's more commonly used for movement in bacteria are flagella. Nope, go back. I thought there was one more thing on there. There's not. Oh, it didn't work. I had a video link of moving bacteria and it didn't come up. My link isn't there. Oh, science. Technology. Okay, so about half of bacteria have some ability to move. So they have flagella, about half of bacteria have flagella. When we talk about taxis, taxis is just the fancy word for movement. Yes, and if you Google bacteria taxis, you get all kinds of information about taxis. And that did not even occur to me when I was trying to find a video about taxis. I tried in bacteria taxis, and I got all these things about taxis. I was like, why am I getting this about taxis? I was like, oh, because it's spelled the same maybe? I don't know. It's a science problem. So you can have different kinds of movement, different kinds of taxis. So you can have positive taxis, which is moving towards something. Or you can have negative taxis, which is moving away from something. So this is movement towards or away from something, from a stimulus. So we could talk about things like chemotaxis, which is moving towards, positive would be moving towards a specific chemical stimulus. Negative would be moving away from a specific stimulus. You can have phototaxis, which is moving towards or away from what? Light. And again, some bacteria are responsive to light and some bacteria are not responsive to light. So some flagella have one bacteria, like you see in this picture. I have one flagella, sorry. Some bacteria have one flagella. I said that. Some flagella have one bacteria. That's also true. But some bacteria only have one flagella. Some have lots of flagella. Some will have them at both ends. And some have really crazy long ones that look like some kind of hair. covering the whole organism. These are different than eukaryotic flagella. I don't know, did you talk about eukaryotic flagella last semester when you talked about like the cytoskeleton and the 9+2? No? Okay. We will probably talk about flagella again with eukaryotic flagella when we talk about protists. So these are different flagella structurally than a eukaryotic flagella, but they're similar between bacteria and archaebacteria. Archaea. They used to be called archaebacteria, sorry. So they're similar in structure between the prokaryotic. All the prokaryotic flagella are similar, and they're different from eukaryotic flagella. There are differences between prokaryotic versus eukaryotic. The prokaryotic flagella are thinner than eukaryotic. That's an I. They're not covered with a plasma membrane. So, and the eukaryotic cells, the plasma membrane actually covers the flagella, but that's not true in prokaryotic cells. And their structure is different, and the way they move is different. So this is showing you a drawing, and this is showing you an electromicrograph of it. I think this microscope picture, I just think is weird. It looks very mechanical, but it's a part of a bacteria. But this is like a motor. So this is the motor. What would drive the motor? What drives you?
Energy.
What kind of energy? What kind of chemical energy? ATP. ATP is the fuel for the motor. So it has ATP pumps that pump protons out. That creates an electrochemical gradient. And when the protons move back in, it provides the energy needed to turn this part, which is called the hook. And that will actually rotate the flagella. And they can move up to 50 micrometers per second, which doesn't sound particularly fast, because 50 micrometers is not a lot. But if you like scale that up to humans, it's something like 190 miles an hour. Like comparing like body size to distance that's moved. So they can move pretty fast. I had a good video of it. I'll have to put it on Moodle because I don't know what happened to it. So external structures, we talked about the cell wall structure, we talked about the outer layer of the gram-negative, we talked about the glycocalyx layer that can form fimbriae, pili, flagella. Let's talk about the inside of bacteria. Bacteria, although we said they're simple and they don't have membrane-bound organelles, they're still organized on the inside. So they don't have those membrane-bound organelles to compartmentalize parts of the cell, but they still have structure. And a lot of them have specialized membrane folding that kind of, the folding of the outer membrane can function as almost like one of the membranes of our organelles. So there's, in the aerobic prokaryotic cells, aerobic meaning what? Using oxygen. They can have these folds of the respiratory, that are called respiratory membrane folds. This is similar to the folds in the mitochondria. Remember, your mitochondria has two membranes, and the inner membrane is all folded and has those cristae, and that's where respiration is happening. Same kind of thing. So it's similar to the mitochondria, the inner membrane of the mitochondria. Bacteria that are photosynthetic are obviously carrying out photosynthesis, but they don't have chloroplasts because they don't have the folded membrane. So they have folds in their membrane that are of thylakoid folds, which are again similar to the thylakoid membranes that are found within the chloroplast. So similar structurally, but lacking that outer membrane to make it a true chloroplast or a true mitochondria. Most of the DNA is found in the center of the, or in one section of the cell called the nucleoid region. And again, the chromosome is a single circular chromosome. So it has one circular chromosome in this nucleoid region. But you know from lab, we can also introduce, or bacteria can take in plasmids, which are smaller circular pieces of DNA. So they're self-replicating, meaning they replicate separately from the bacterial chromosomes. So it's a separate process of replication. A lot of genes for antibiotic resistance or toxins are found on these plasmids, and we can manipulate them in research like we did in lab by adding things like the green fluorescent protein gene. Again, prokaryotic cells use a similar way to express genes with transcription and translation, but proteins that they produce are somewhat different than eukaryotic cells. So again, if we can target those proteins that are specifically only found in prokaryotic cells, that's a way that we can treat bacterial illnesses without harming a host. So if we can find ways to target proteins that are specific to a prokaryotic cell, then we are not putting our own cells in danger when we treat those bacteria. Okay, so we've talked about structure.
Is there a limit to how many plasmids a bacteria can take up? Because there's three pictures.
I would be sure there is some kind of limit, but it probably varies on bacteria size too, because bacteria have their own sizes, so I would guess a bigger one could take in more, but so I don't know of a hard number that we can say, yes, they can take in 17 and that's it. But I would guess there is some limit because there's a limit in space and raw materials to keep replicating and expressing those genes. Okay, so bacteria also have to reproduce because they're carrying out all the functions of a living thing. They have to be able to reproduce. And so there's a couple different important things that we have to talk about with reproduction. Binary fission is the way bacteria reproduce asexually. So binary fission is just splitting. So the DNA replicates, kind of like mitosis. The DNA replicates and the cell splits in half, and each daughter cell gets new DNA. Okay, it's an asexual reproduction process because you're making exact identical copies of the cell. It's not a way to increase genetic diversity, but genetic diversity is really important in bacteria. And there are different ways that bacteria can become genetically diverse. It can take a bacteria as little as 20 minutes to replicate, to reproduce. Okay, so if you start thinking about you getting a bacteria in your body, and every 20 minutes it doubles, and every 20 minutes it doubles, it doesn't take long to get a significant number of bacteria. But because, again, this reproduction process is asexual, it's not creating diversity of bacteria. So how do we get genetic variability? The main way is through mutations, which we didn't spend a lot of time talking about last semester. But when DNA is replicated, sometimes mistakes are made. So that's a mistake in the replication process. And again, because bacteria are replicating so fast, those mistakes can build up pretty quickly. So even though mistakes are pretty rare, sorry, because everybody proofreads, it's still, if you're copying yourself every 20 minutes and you're doing this over and over and you get millions of bacteria in a very short period of time, if 2 bacteria mutate, have a mistake, and there's 1,500 genes, or 1,500 genes, and one has a mutation in this one and one has a mutation in that one, mutations can build up pretty quickly in a population. So the rate of reproduction and the rate of mutation is a way to introduce genetic diversity into a bacterial population. Genetic recombination also helps. So genetic recombination is combining DNA from 2 sources. And there's 3 ways that this can occur. Transformation, transduction, and conjugation are ways that bacteria can combine DNA from different sources. So transformation, that's what we did in lab. So this is taking up DNA from the environment. So we literally introduce DNA, but there is DNA just plasmids floating around in the environment. And so bacteria can pick those up, they can transcribe them and translate them, and incorporate them into their own genome and pick up new genes that way. Genes for antibiotic resistance. Transduction is similar. It's DNA moving from one bacteria to another via a bacteriophage. What's a bacteriophage? It's a virus that infects other bacteria, but they can also carry information from one bacteria to another. So genes are carried from one host to another via a phage bacteria. And that's called transduction. And the third way is conjugation. And this is between 2 bacterial cells that are connected by a pilus is the singular of a pili. We shouldn't say a pili, we should say pili and a pilus. So they're temporarily joined by a sex pilus, and they can send DNA from one cell to another. Is this sexual reproduction? It depends on who you talk to. So some people say it's sexual reproduction, some people say it's not sexual reproduction. And here's a picture of it. Here's the bacteria, here's the pilus. It's a temporary connection, and bacteria can exchange, it's not exchanging of DNA, it's bacteria going one way. DNA, going one way. It's a one-way street, it's not an exchange, so it's not one gives one some and one gives the other some. And antibiotic resistance we've already kind of alluded to. Antibiotic resistance can come by a change in the chromosome of the actual genome of the bacteria, or uptake in a plasmid where bacteria just acquire a gene that makes them resistant to an antibiotic, so that they are no longer susceptible to that antibiotic. So their bacteria are prone to mutations because of their rapid reproduction rate. They have genetic diversity because of the mutations and because of genetic recombination. But the reason that we're not totally and completely overtaken by bacteria is because their reproduction is limited by things like the amount of nutrients, by competition from other species, by the fact that our bodies are trying to fight off bacteria so that we're not completely taken over by bacteria.
Can you run it back one more time for the antibiotic resistance?
So antibiotic resistance can come by a mutation where the cell has a mutation that makes it resistant to antibiotics, so it's a part of its actual genome, part of its chromosome, or it can come from a plasmid coming in, like we did in lab. So competition, predation, nutrients, all those kinds of things will limit reproduction rate so that the world is not taken over by bacteria because of their rapid reproduction. Bacteria are also metabolically diverse, meaning they can use different ways to get energy, and lots of different ways to get energy. So bacteria have different oxygen requirements. Some are obligate, meaning they have to have oxygen. They're obligate aerobes, or they're obligate anaerobes, meaning oxygen kills them, and they can't have oxygen. Facultative means they can do it if they have to. So there are bacteria that are what we call facultative anaerobes. They would rather have oxygen and they're happier when there's oxygen. But if there's no oxygen, fine, they can do something else. And anaerobic versus aerobic. Anaerobic meaning not oxygen, aerobic meaning oxygen. And aerotolerant bacteria are okay with oxygen, but they don't use it. So they can be in the presence of oxygen. Oxygen doesn't kill them, but they don't use the oxygen. Bacteria need nitrogen, just like you do. What kind of compound in your body has nitrogen? Okay, there's 4 main biological macromolecules. Maybe. Yes, it would. What category is that in? There's carbohydrates, proteins, lipids, and nucleic acids. Which one needs nitrogen? Both the DNA and RNA, so nucleic acids and proteins need nitrogen. Okay, so bacteria need nitrogen because they need it for amino acids to make proteins. They also need it for nucleic acids for the nitrogenous bases. There's tons of nitrogen in the atmosphere. The air concentration of nitrogen is around 71% of the air is nitrogen. But it's not usable nitrogen. It's inert, it doesn't react very easily. So we get nitrogen by eating stuff that has nitrogen in it. Most bacteria get nitrogen by eating stuff that has nitrogen in it. But some bacteria are able to fix nitrogen. So they take nitrogen from the air and they make it into ammonia, which is a solid that they can use. This is very useful for them, but it's also a useful process for us. Okay, nitrogen fixing bacteria are really important in the nutrient cycling, moving nitrogen into the soil for plants to use. Can you not read it? NH4. It's ammonium. Ammonia is NH3. Sorry. I might have said ammonia, but it is ammonium. Some of you must have taken chemistry. So this is really important in nutrient cycling and for bacteria, for us to be able to access nitrogen. For carbon, again, a lot of bacteria can make organic compounds through the process of carbon fixation, also known as photosynthesis. So again, they can get carbon by consuming it. So taking in sugars, carbohydrates is a good way to get carbon. That's how we get our carbon. But some bacteria, like cyanobacteria, which you looked at in lab, can do photosynthesis, which is carbon fixation. And bacteria can use a lot of different energy sources. So photosynthetic bacteria use what as their energy source? So they use the light. There are some bacteria that are chemosynthetic. So they use chemicals, often hydrogen sulfide, like in ocean vents. So they're still autotrophs, meaning they're making their own energy, but they're using hydrogen sulfide instead. Or some are what we call photoheterotrophs. So they still use light for energy, but they don't use carbon dioxide as their only carbon source, so they still have to take up carbon to do photosynthesis. And then we are, like some bacteria, chemoheterotrophs. We take in what we need to get. We use chemicals to get energy, and we take it in from other sources. We don't make it ourselves. And we are done, aren't we? Okay. I will see you on Monday. We might talk about bacteria for one more slide. Or two. And then we'll start chapter 28.